This is module 5.2.2. This is one of our guest presentations for today. We have Huijing Wang presenting from Carnegie Mellon University on some work that she's done recently in CompuCell. So Huijing is a postdoctoral researcher at Carnegie Mellon University. Her research focuses on mechanical forces and collective cell migration, among other things. And today she will be presenting a recent CompuCell paper that she published on protrusion-force and cell-cell adhesion-induced polarity alignment governing collective cell migration modes. I believe Huichang was a previous workshop attendee. It may be, I don't remember which year it was, but
Yeah.
was it back from when we were still doing this in person?
I think it was COVID year, so
No, okay.
everything is remote.
Yeah, everything's COVID years where it was like all one big year, so it's hard to remember when exactly. But Huichang is a previous workshop attendee. She's coming back to present her newly published CompuCell work. Just as a reminder, this session is being recorded and will be distributed later on YouTube. We are not currently being live streamed, but the session is being recorded locally. So just keep that in mind. And I'd like to thank our funding sources through the NSF, as well as our support networks through the IUB Biocomplexity Institute, as well as the OpenVT and Glimprint initiatives.
So thank you for the introduction. I'm very glad to be here to present my my work on protrusion-force and cell-cell adhesion-induced polarity alignment govern collective migration modes. So this is a work I did with within Jianhua Xing lab. So from this cover, you already can see that the tumor model I generated with CompuCell3D have a lot of resemblance with what we have in the experiments. So why are we interested in tumor migrations? And then here I'm showing two pictures of T47D. It is main characters in the experiments. It is a breast cancer cell lines that we were using in this paper. And the reason we are interested in tumor migration is firstly, it is very lethal. So typically, tumor metastasis happens at stage 4 cancer cells, where you can't completely get rid of all the tumors, and then the tumor shed cells into all places. It goes into bloodstream and spread to distant organs and reoccur. So it is very hard to treat tumors when it starts to migrate, and then also different migration modes may contribute to invasive invasion efficiency and metastatic potential. We also are interested in the tumor plasticity and the mechanism underlying hypoxia-induced migration. Here I'm showing an example. When looking at the ductal carcinoma, we found it does not have effective treatments. So basically, it have local reoccurrence and metastasis, and it's hard to predict the patient at risk. So in order to understand the tumor metastasis, our collaborator, Dr. Shang lab, developed this very cool hydrogel device. So on these hydrogel devices, you have lots of microwells. The diameter may vary. The larger diameter is 600 micrometers, and smaller ones are 150 micrometers. And those, this hydrogel device allows you to seed specific amount of tumor cells into each well. And then using this device, you can also accurately control the microenvironment for those microtumors. Over the course of a few days, those seeded cells can grow into microtumors. In this accurately controlled environment, you can mimic the in situ disease development. And if you're interested, I listed our collaborators' papers. So in previous research, we found that if seed cells into these large microwells, which have the diameter of 600 micrometer, over a few days, after a few days, the microtumor can start to migrate. We found that this type of larger microtumor has intrinsic hypoxia. This intrinsic hypoxia induce expression of secretome that leads to directional migration, as we shown here. So we want to understand the underlying mechanism for this metastasis. So we use a conditional medium, which is the culture media collected from this large microtumor. And then we added into a smaller tumor. So this, in the lower part, we have a smaller microwells. In each microwell, the diameter is 150 micrometer. And then we found that without adding secretome, the small tumor, they keep static. They're not migrating at all, even after the same 6-day period. So, and also we use a RNA microarray to confirm that there is no hypoxia-related genes are expressed in the tumor. So there is no hypoxia in small tumor. Basically, the oxygen is permeable to the center of the small tumors. And then we added the secretome of 600 microtumors into the 150 small microtumors. We found that almost all of those tumors start to migrating, but in a very different way. We observe this type of radial migration patterns. So this is interesting. For the same cell line, we observe directional migration and radial migration patterns. To understand the mechanisms, we did the RNA microarray analysis, which is shown here. We found interestingly that in large microtumors, the cell adhesion components are upregulated, while in 150 smaller microtumors, those adhesion components are downregulated. And then in large tumors, those protrusion and protrusion for polarity generating components are downregulated, but it is upregulated in the small microtumors. This leads to the question, so what determines how group of tumor cells move together, and why can the same tumor switch between different migration modes? Yeah, with this question, we start to build our minimal model with CompuCell3D. Mainly, it have 5 components. We can go through it one by one. First, it is secretome diffusion field. Basically, the small microtumor migrate because we added secretome to it. So so our hypothesis is that secretome have this diffusion field that it slowly diffuse into the tumors. So here we have a diffusion term, coefficient D, and it's diffused into each pixel of that cells. And those cells take up the diffusion signals. And then for a single cell, for each single cell, there are multiple pixels inside it. So our uptake of secretome signal is simply the summation of all the secretome signal for each pixel, summation divided by the total area of that cell. So we have a density of secretome signals for that single cell. And then this is a constant coefficient. And then we know that for single cell, it have uptake a certain amount of secretome, and then we have these R components. In tumor cells, there are many different components that can generate protrusion force and reorganize cytoskeletons. The most famous ones are Rac and rolls, so we use R to represent those protrusion force generating components. We know inside the cells, there are always some amount of basal expression of those protrusion generating components. So it is represented as B, and then this S is from the secretome uptake. Of course, we have this dilution term, and this one gives us the dynamics of the single cell protrusion for generation components. And then this is force vectors. So our hypothesis is that the more of the protrusion force generating components inside the cell, the stronger the protrusion force should be. And to generalize these processes, we use a Hill function. And then with these settings, we can have our model energy for protrusion. The next one is intracellular adhesion junctions we simulated with the FPP adding in the software. And then this is a tumor shape constraint, which is inherent. Basically, we can find the cell area and the parameters. This is surface properties. So basically, this is saying that in our model, the cells tend to stay with another cell instead of running away. So I believe you have already familiar with all those cell pot model energy terms. So combining those 5 components together, we have our model energies. Then we can have a look of the simulation. So this is the initial steps. There's no secretome added, so there's no protrusion force generated. And then so in the first 19 MCS Monte Carlo steps, it is relaxing the models. So there's after 90 steps, we have expression of the secretome. Here we can see that this is radially migrating tumors, and this one. Again, first 90 step, it is without secretome, so there is no protrusions. The color regime here showing the protrusion force of single cells. After adding secretome, we can see that the signals diffuse into the tumor cells. So at the boundary, it has the highest concentrations of the secretome, so it generate the strongest protrusion force. And this is a directional migration. So these are the typical simulation snapshots we have, and use this simulation we can obtain some geometric features. For here, I'm showing convex area. The convex area is for this complex geometric shape, we're looking at the convex hull, total convex hull area. And then we can see that for radial migration, the convex area keep increasing, but for directional migration, the convex area keeps smaller, which is what we expect. And then also the tumor displacement is just the distance of the center of the mass for that tumor compared to its original position. We can see that if the tumor is migrating directionally, the displacement keep increasing, but for radially migration, the tumor center of a mass keep near the original locations. So we definitely can observe those tumor features by eye, but then we want to systematically analyzing these features. We can use unsupervised clustering for this. We collected 19 different geometric features, for example, like what we have convex area, tumor displacement. And if you're interested, you can look at supplementary. There are definitions for all those geometric features. And here in the left panel, I'm showing the distribution of those geometric features. Those geometric features make sense when you're doing clustering. So first, you can do a principal component analysis. So to clustering the migratory tumors and non-migratory tumors. And then the second step, you use the same methods. First the PCA and then doing k-mean clustering. You can cluster the radial migration and directional migration. We verify our clustering by looking at the actual tumors we have here. We found that the cluster method works great. In the panel D, we have non-migrating tumors where we can see that the center of mass overlay well with the original. And for directional migration, we have very good directional migration, and radial migration is more sparse. This method allows us to process all the simulation data in batch. So here I did parameter scans for each set of adhesion strength lambda FPP and protrusion-force F. For each pair of parameters, I did 50 repeats. So basically, for each block, you're looking at the overall results for 50 simulations. And then for each block, the different color area represent the probability of that condition of the migratory mode in that condition. So the reds are non-migratory, the blues are radial migrations, and the greens are radial migrations, and the blue are directional migrations. From this phase graph, we can see that when the protrusion force is large and the adhesion is small, we mainly have radial migration mode. And if we have large protrusion force, but also larger adhesion, we have a directional migration mode, but also non-migration cases. And if we have smaller protrusion force, and it doesn't matter if it's a strong or weak adhesion, we have non-migrating tumors. I think it's interesting if you look at single cell polarities in panel B, the radial migration, the cell polarities are very random. The spectrum ranging from negative pi to pi, but for directional migration, the polarity of tumors are very much aligned. So with this observation, we come up with an order parameter phi. So this phi is a tumor polarity term where the upper part is the vector summation of all the polarities of single cells divided by the total norm of the polarities. And then, so this one calculates one time point of the polarity of the tumor. And then if you divide it over time, the whole time course, you can have an average temporal polarity. And then for cell polarity, it is looking at a variant how the single cell spinning comparing to its average polarities. So I plotted this heat map in CND. We can see that for average tumor polarities, when adhesion is high and the protrusion force is high, there is very high average polarities. And for cell polarities, it peaks whenever there is a large protrusion force, very small protrusion force. And then in panel E, it is more obvious that the three migratory modes are clustered in three regions on this phase diagram. If you have higher polarities and moderate cell polarities, you can have directional migration. And then if you have a small average polarities and small cell polarity, you can have a radial migration. For non-migration cases, you can have a very large cell polarities, but the average tumor polarities is very minimal. So with this parameter scans, we have lots of information how the microtumors mode going to change. So we want to verify our findings by tuning the parameters. Here we keep the adhesion the same, and we want to perturb the protrusion force. If we found that if we increase the protrusion force, the radial migration does not change the migratory mode, but instead it is become more spread out. We can see that with increasing the protrusion force, we have a larger convex area, and we have a larger cell displacement. So does it consistent with our experiments? We designed this experiment by inhibiting myosin. So biblistatin, it's inhibit myosin. So myosin is considered a protrusion force generator. If we inhibit it, it's supposed to reduce the protrusion force. So indeed, we observed that if we inhibit myosins, we observe smaller migration distances. This is comparing to this control panel and biblistatin. So this verified that protrusion force does play an important role on the migrations. And then we want to look at adhesions. So we found that strong adhesion promote directional migrations. So we keep the protrusion force the same, but keep increasing the adhesion strength. So our lambda FPP increased from 50 to 400, and then when the adhesion is weak, we found all cells are radially migrating. After we increasing the adhesion to a moderate level, we found that we can observe both directional, radial, and non-migration tumor modes in the same conditions. If we keep increasing the migration, if we keep increasing the adhesion coefficient, we found that the cell either directional migrating or non-migrating. This is also consistent with our experiment. AKT2 is a so in our experiment we found the PI3K AKT2 pathway is differentially regulated in directional and radial migration modes. And the AKT2 function is less known and may induce migration mode changes. Indeed, we found that in our control that all the tumors, they are migrating radially. And if you use CCT, this small chemical component to inhibit AKT2, we're going to have a directional, radial, and non-migrating tumors. This is consistent with our simulation where all three migration modes coexist. So in summary, this is also my title. So the protrusion-force and cell-cell adhesion induced polarity alignment govern collective migration modes. So and also CC3D is a very powerful tool. It extends our understanding and perspective towards cell and tissue activities, together with other analysis methods such as unsupervised learning here, which is a type of basic machine learning method. We can better visualize and understand the underlying mechanism of emergent phenomena. I think I'm very lucky that our question is particularly interesting and fundamental, and CC3D tool is so powerful that it can capture all experiment observations and also computationally analyzable. My future directions is although this model is doing generated using CompuCell3D, but it's actually very much 2D modeling. So I want to incorporate the 3D components to better understand the tissue development. And also, I think it will be interesting to implementing intrinsic cell features such as cell growth, division, dormancy, and cell death. And also, I want to targeting gene expression profiles for abolishing tumor migrations. This may shed light on the tumor treatment. I want to acknowledge Dr. Jianhua Xing, Dr. Catalina, all my collaborators, and also I'd like to thank CC3 developer, Dr. James Glazer, Dr. James Slanka, Dr. Giuliano, and Dr. T.G. Siegel. And also I want to acknowledging grant and funding from NIH.
I have a small, naive question that I've been wondering for a while. What exactly classifies something as a microtumor? Is there a standard for how small does it have to be micro?
I think in our experiment, the microtumor, yeah, it's basically meaning that those tumor growing from those microwells. Currently, we have two sizes. So 600 micrometer is a slightly larger version where it have intrinsic hypoxia, and the smaller one is 150 micrometers.
Yeah, I was just trying to get a sense for scale there. Any other questions?
So in my shared folders,
Sorry?
folders, there is a folder called the tumor-model FPP force. So this model contains CC3D model that I'm currently sharing on the screen. And then You can see this current parameter is for adhesion strength is 200 and the force is 300, and then the cell, the tumor shows a migrating behavior, radial migration patterns. And you can toggle those. So now I'm showing the secretome. You can also look at the force on the force, and this is the overall force. You can look at force on the x component and the force on the y components. And this is F force combined. You can also look at the secretome induced protrusion force generation component expression. Here I marked it at Rac. And then this is polarity, combined polarity. But if you look at the x components of the polarities and the y components, and then you can keep running. You see that over the time course, the tumor changing, each cell, they are changing their polarities. We can change different parameters. If we have a smaller FPP and a larger protrusion force, we're expected to have a radial migration. So this is relaxing step, and then after secretome add-in, we're going to have this radial migration patterns. And then if we tune our parameters to have a strong adhesion, and rerun the simulations. We can look at the polarity X components. We can see that those components have very consistent directions. So basically, you can run this simulation for longer. Currently, in these simulations, I have a total simulation step of 390. So I can start by going over the code. So this is a CompuCell3D model. Here I'm defining the simulation run 319 steps. And then here I'm defining the cell types. There is two types of the cell. One is medium, one is cells. And then I define constraints for volume and surface. Because our model is 2D, it's basically the parameter and the area. And then I use the neighbor tracker so that I can tell for each cell what are the neighbor cells so that I can calculate the polarity alignment. Those contact energy basically meaning the cell tend to stick together with cell better compared to cell to the medium. And this one using the focal point plasticities. I also use the diffusion solver FE for simulating the diffusion secretome field. And at the very bottom is I'm initializing all myself in a blob initializers. So you can define the radius of that blob and define the width and then define what type of the cells. So here is a overview of steppables that I'm using. So basically for each step of Monte Carlo steps, it will looping through these functions. So it will first look at rack, and then like for every step, it will run this migration rack steppable, and then it will run one step of ODE calculation for each single cells. And then it's for every 10 Monte Carlo step, it will recalculate the focal point plasticities. The reason for that is that we think cytoskeleton reorganizing and changing of a cell-cell adhesion strength is actually time-consuming because you need to um, uh, have all those proteins ready and then produce a correct structure. So this is a slower processes. And then the next is update the ODE. So let's go to the steppables. Here are the two stage of the simulation between in first 120 steps we're relaxing the model as it is, and then at 210 steps we are adding secretome. Those are inherent parameters. We define initial rack level inside the cells, and we say that the rack components inside cell has to above 40 to be effectively generating the protrusion force. This is our single cell R dynamics, where you have the generation, the production of R, and you have a degradation of R. So for each step, I use a cell dig to collect all the all the cell-related property, for example, the XY positions and its past XY positions. The past XY positions helps me to calculate the cell polarities. And these are calculating the cell polarities. So basically, you're using current XY position minus the XY position that 5 steps ago. So basically, the polarity is reflected based on the displacement history. And so after adding secretome, I use this function to calculating the total secretome signals inside the cells. And then you can see I divided by the cell volume to get a secretome density for each single cells. And after I added the secretome, I keep updating the secretome. I keep updating the secretome density inside single cell for each Monte Carlo steps. So this is this ODE is steppable. It has two functions. One function is that I can look at all the cell properties, which is defined here. So all those property defined here, you can observe it in this manual. So it is corresponding to all these like secretome XY polarities, force XY components, total polarities, and yeah. And this is antimony. So basically, you can for each cells you can add an antimony module that allow each cell to simulate the R uptake, and then we can have the protrusion force generated differently for each single cells. This is a focal point plasticity add-in. Here is where I define the coefficient for the adhesion strength. This function updating the polarity based on the neighbors. So basically, we think because there is a cell-cell adhesion between neighbors, the neighbor polarity actually impact this cell polarity. So it is a so you can see the polarity is actually a fraction of its own polarity with a fraction of neighbor polarities. And then the vectors of xy is normalized to 1. And then the final part here is basically for each Monte Carlo step, I'll write a file showing all those parameters and the dynamics of the model. Is there any questions about this model?
Yeah, this model includes a few things that we'll be talking about next week in our advanced topic sessions, but if you have any questions about them right now, this is a good sneak peek, so please feel free to ask. Yeah, Levi asked, what is antimony? Antimony is a network modeling specification language that you can implement through a Python package called Tellurium. In CompuCell, you can use tellurium/antimony on its own, but you can write chemical signaling models in antimony and then load them into cells in CompuCell. And we'll actually be talking about that first thing on Monday and Tuesday about how to do that to add chemical signaling behaviors to your simulation.
Yeah. Yeah, antimony is very flexible. It can use any SVML, um, like a Gillespie simulations incorporated into single cells. For me here, I'm using a very simple model here where it's just a simple production and diffusion of R of R. But you can always have a very complicated dynamics for single cells. So for here, I want to show that. So every time you run a simulation, it will generate this type of result folders. For me, because for each Monte Carlo steps, I generate text files. This is might not be the most efficient way to record data, but I can have this type of data sheet where each column is a different thing. So for example, the first column is cells ID and second one is X position and Y position for each Monte Carlo steps. And then using this data, I can analyze the geometric features of the cells. So basically, this is a example of analyzing geometric features. So it is in the shared materials in the feature calculations. In the feature calculation, you can see there are lots of functions. It's read the time theory of our simulation files, and it's look at the polarities, and also it's calculating parameters. So basically, all 19 geometry features can be calculating through this function. And then if you're running analysis for your simulation results, for example, here I'm using one of the analysis I've previous run. So this analysis I actually run for 800 steps, so that this is a directional migration mode case, and then I ran it very long time, so all the cells migrate to the boundaries of my screens. And then after I running it, so this one is a velocity correlation. You can see the velocity correlation at this final step is almost 1, and at this final step, the cells are all at the boundaries because this is a directional migrations, and then this is the original position of the tumor, and this is the beginning of the simulation. We can see the tumor initialized in the center, and the velocity correlation of the cells is everywhere. So this is the beginning, and this is the end. So basically, with this analysis, it automatically I can generate this type of results. For example, I can have my folder path, file path here, and I can have a parameter scan here, and then the components of the rack inside the single cells, and these are all the geometric features. For example, tumor cell displacement, tumor displacement, and then all native features. And those are if you combine these feature analysis data for 50 repeat, you're and then doing clustering, you'll be able to you can then get this type of phase graph. Yeah, any questions?
I've enabled the ability for people to unmute, so if you'd like to unmute and ask questions at this point, feel free. Sounds like there are no questions.
Okay.
Does everybody have the code downloaded and were you able to open it on your end? Margo says yes, good. Levi, yes, okay. So did you have another activity lined up, Huichang, or?
Other activities, I would suggest that so simulating larger tumors. Currently, I'm using a minimum number of tumor cells and using a small panel. And so it's only reflecting the initial state of the seeding in small microtumors. Yeah, so it might not reflecting the true dynamics of larger tumors, and also it does not contain any hypoxia mechanism since we're focusing on the small tumors for now. But I think it's always interesting to look at a larger tumor and the dynamics. Another thing is to simulate with cell growth and division. One thing I think it would be interesting is to look at to change the p-frac scalar, which is the beta here. This coefficient basically defines how much the neighboring polarities will neighbor cell polarities will has impact on these single cells. The larger of neighbor alignment coefficient, the better the cells are aligned. For example, I can have this become 0.1 so that cell are not very aligned. Leave it and because cell polarity are not aligned at all. There is a larger chance for the tumor to become radial migration. So actually, if we changing the beta, like local how the local cells are aligned, we can change the dynamics a little bit. We can see that if we have no no alignment between local cell due to adhesion, we have a larger region for the radial migration. But if we have we have a very strong alignment between neighboring cells, we have a smaller region for this radial migration and a larger region for the directional migration.
That is interesting. Is anybody giving that a try on their end? You can share a screenshot in the chat if you've got the simulation running. Show what yours looks like. Did you end up with the radial cell rave, like in the one Huijing just showed?
I also have all the corresponding, for example, the energy equations with corresponding the code here in the shared slides.
Hmm. Yeah, that's actually very useful. That's something that we didn't talk about too much in the workshop is the how we tie our mathematics to the specific plugins that we use. So let's, if you can go back to your activities slide,
Mhm.
let's pick, so for those who have the simulation running, in order to simulate with cell growth and division, what would you have to add? We've got a little bit of time. Maybe we can spend 10 minutes on adding that to see what it looks like. Does anybody want to give that a shot?
Oh,
Ragu has it running. Is this just the base simulation, Ragu? Decrease the scalar value to 0.005. Oh, the beta p fraction.
Did anyone try to simulate a larger tumor?
Or if anyone wants to try running a larger tumor now and share their results. One question here, would changing the neighbor order just make the cells stick together more?
I think it has minimum effect.
All right. Well, any last questions? I know people are probably getting tired at this point. It's end of the week. Any last questions? Anybody else running the simulation have anything they want to share or ask about? All right. Well, I think if not, unless you had anything else you wanted to show, which one we can probably wrap up there.
Okay.
Um, but we have her materials in the student materials folder if you'd like to take a shot at this. Looking at her simulation and maybe adding a couple of these extra things like simulating larger tumors or adding cell growth and division maybe over the weekend as a practice exercise. Um, you have access to all of those materials. So, all right. Well, I think with that, we'll thank Huijing Wang for joining us today. This is a pretty interesting and kind of big simulation with some new ideas in it. So I think people just need a little bit of time to wrap their heads around it. All right. So thank you, everybody.